Good evening. I would like to say thanks to Madam Sulagna and Dr. Prashad who invited me here. And I will start my talk right away. And I notice my talk, whatever you people have listened, it is little or even entirely different talk. I'm going to focus on the how we can improve the crop production in presence of different kind of stresses going on, either in the Himalayan area or in the other areas. So I will describe you in short little bit about the stress. Sorry. Then I will tell you different approaches, microbial approach and some other approach, how we can improve the crop through genetical modification or without the genetical modification. What happened now, you see, in response to different stresses which are mentioned, there are abiotic stresses and the biotic stresses. Abiotic stresses mentioned here and biotic stresses. Because of the negative impact of these stresses, the crop production is getting decreased. Actual potential is this much, but because of the negative impact of these stresses, the production is going on this much less. So how we can improve the production while in presence of the stress? That's I'm going to focus on that. And on the other hand, our population is increasing. In 2050 is going to be close to 10 billion, and from 6 to 10 billion. And production is not going on with that increase. So in future there will be a problem and the food security will be in the danger. So that's why it is very important to develop the stress tolerant variety so that we can cope up with this negative impact. As you can see in the NASA picture, wherever you see the red spot, the land of this area is already affected with the salinity, too much salt. So either there is no production going on or production has reduced a lot. Similarly, in response to the drought stress, you can see red patches, so production has decreased a lot. So in combination between the salinity and drought, which are the major negative factor for the abiotic stresses, the production decreases a lot. And you can see it is very difficult to grow some crop in the very drought condition and in the soil which has too much salt on that. But through genetical modification, through biotechnological approaches, there is a possibility that we can grow that with some modification. There are several approaches. The approach which I am going to focus here right now is the microbial approach. There are some beneficial microbes which helps plant to grow under the stress condition also. And one of the beneficial microbes is the Periformospora indica. It's a multifunctional fungus. It has been discovered from this university by Professor Ajit Verma in the desert area, and this is a kind of a magic function. It provides a lot of magic role to the plants and helps to the plant in tolerating the salt, but tolerating the different kind of stresses and helping the plant to grow much better and better. But the mechanism is still not known, and that's what we have focused on the mechanism part also. So the approach that I'm going to describe, non-transgenic approach using the microbial approach, through microbial approach, how we can do the non-transgenic approach or through transgenic approach. Through the non-transgenic approach, you can see if we inoculate that fungus, the beneficial fungus, and then grow the plant, and then you can see the difference in the brassica without inoculation and with inoculation, the growth is little better in some tomato plant, in the tobacco plant, in the rice plant. In presence of inoculation of this particular fungus, we are getting little better growth. Some more example you can see here in the Pisum sativum without inoculation, with inoculation, and some other plant also you see, and some recently we have done this in onion. You can see onion production is getting increased in presence of after inoculation with the P. indica fungus. And some medicinal plant can see, some medicinal plant also you can see In medicinal plant also you can see the production is going on more, so that we can extract more drugs from those plants also. And you see the turmeric, you can see without inoculation, with inoculation you see the more better growth and more production you can see. And artemisinin, which are recently been going to use in the malarial drug, as one of the malarial drug, you can see the difference between the growth of the plant. These are the non-transgenic approach. So, but how we, in order to understand how they are going, so we have how they are doing it, so that we have to understand the mechanism also. For mechanism, we wanted to know whether any genes are involved for this kind of beneficial role in the plant by this fungus. And through that mechanism, we have studied through genetical approach. We have isolated several stress tolerant. The technology, because I cannot describe, because I feel the audience is not, may not understand until you have a lot of background on that. But we have developed a technique so that we can isolate several genes which are playing important role in presence of stress. Stress tolerant genes we have isolated from the same fungus, and then we have cloned those genes, sequenced those genes, and then there are a list of those genes here mentioned. But today I will focus on particular one gene. This is cyclosporine, and there are so many genes which are we have isolated. They are all stress tolerant genes. They have some kind of role in stress tolerance. So we have cloned that several genes, but cyclosporine gene we have introduced in the tobacco as a model plant, and that technique also we have developed in our own lab, how to integrate the gene in the plant. And finally we have developed a transgenic or genetically modified plant which has the fungus gene integrated in the plant. And then you can see we have tested here for salinity stress tolerance. You can see we have cut the leaf disc from the leaf of the transgenic or non-transgenic plant here and grown in a water as well as 200 millimolar salt solution. And you can see grown in a water is a wild type plant as a vector control, which is transgenic, but there is no that gene. And you can see they are becoming yellowish here. That means they are not tolerating the salt, and the transgenic line are tolerating the salt. Similarly here, the next generation we have grown this in presence of continuous presence of salinity stress, and then collected the seeds, and next generation we have grown in the seeds were germinated in presence of 200 millimolar salt and water. The wild type plant as a vector control and 3 transgenic lines, you can see they are growing nicely here in the water, but transgenic lines are also growing much better as compared to these in presence of 200 millimolar salt. That means transgenic lines are tolerating the salt. In presence of continuous salt, the plants are growing. And then we wanted to know what happened in the matured stage of the plant. When the plant becomes mature, at that time we have added the salt later. And then you can see after adding the salt, the wild type control, where our gene has not been gone, and the transgenic approach, transgenic plant, but not with our gene, only the vector control, you can see they are dying. But the transgenic plant, where the cyclosporin gene is there, they are survived. So that means we have developed a salinity tolerant plants using the beneficial fungus gene. There are some other approach. In short, I will describe you. That was a microbial approach. Now I am telling you non-microbial approach from plant gene to the plant gene. You might have noticed we all have all kind of genes, but all the genes might not be expressing all the time. Similarly, what we have done, we have isolated one very important gene which is known as DNA RNA helicases, which play a very important role in unwinding the duplex DNA or destabilizing the secondary structure in the RNA, which are very important in order to have some other functions going on, like DNA replication, repair, recombination. For that, transient single-stranded stress is very important, and that is provided by the DNA helicase enzyme. And that we have cloned the DNA helicase gene. And now I am telling you the example after several years of after research. I am showing a single gene that which is known as plant pDNA helicase 45. Single gene provide different kind of stress tolerant in many different crops. Here you can see the gene, this is the helicase gene which opens the duplex DNA, which we have published in Nature, that is long time ago. And first we have introduced in the tobacco, you can see the transgenic tobacco, genetically modified tobacco plants are growing in continuous presence of stress, salt, while wild type can't grow. Similarly, you can see here rice variety are growing, different rice variety are growing, and the groundnut also you can see the difference, wild type versus more yield, even in the drought condition you can see more yield. You are giving less water, 50% less water, but you are getting almost double yield also. So that means it's providing salinity as well as drought stress tolerant in a different crop. Here is the sugar cane, you can see in presence of salt stress, they are continuously growing. And recently we have introduced our gene in the onion and chili also. Onion we have not published yet. Recently we have published a scientific report few months ago that chili, when we have introduced the PDH45 gene in that, and chili is growing very nicely in presence of salinity stress as well as in the drought stress, and we are getting more yield also. There is no yield loss. So that's an example of single gene providing stress tolerant different crops. Then I'll show you another example where you see a single gene like calcium ATPase gene function in different stresses also, providing salt tolerance. Same gene is providing salt tolerance, drought tolerance, cold tolerance, and heavy metal stress tolerance here. This is salt tolerance. See that wild type is not surviving, but transgenic lines are surviving in presence of continuous 200 millimolar salt. And similarly here in the presence of drought condition, you can see they are growing, transgenic plants are growing, and in the drought condition also you see transgenic plants are growing in the less water, much less water, while wild type cannot grow. In the cold condition also you can say wild type growth has reduced and later they die. But in this is the experiment was done 4 degrees centigrade in the cold room. In the cold room experiment, see they are still growing the transgenic. And cadmium is a toxic metal, heavy metal, and then that also in presence of cadmium they are tolerating salt. So single gene is providing 4 different kind of stresses. And there are a few more examples, maybe time may not be enough, so I will not go in more detail. But you can see step by step, there are 4 plants here with the control and transgenic. These are the last one, this was the transgenic, these are different kind of control. You can see with the time, and they are all dipped in 200 millimolar salt. And you can see with the time, in 2 days, in 12 days, in 30 days, whatever you see here, only transgenic is surviving in presence of continuous salt. So I will end my talk here. There are so many examples that we can, because of the agriculture, because production is getting decreased, how we can improve the production in presence of continuous stress through genetical modification, which is one of the fastest way, and non-genetical approach also is possible. We can improve the crop production. Thank you very much. And the same approach can be used for the Himalayan and other area also. Thank you very much.